Hello, my name is Nhu-An Pham. I'm a staff scientist at the Princess Margaret Cancer Centre. I manage the Princess Margaret Living Biobank, the PDX core, and today I'd like to share with you some of our work using the PDXPDO patient-derived tumor models to better understand tumor heterogeneity in drug sensitivity and response in adaptation of the tumors. I have no potential conflict to disclose. I like to address the learning objectives of what these models are, how we use them, and where can we access these models. I also like to emphasize the importance of generating models that reflect the disease spectrum, and I describe the use of these models in our non-small cell lung cancer research program that's led by Dr. Ming-Hsiang Chow and Jeffrey Liu at the Princess Margaret Cancer Centre. And also I like to describe how these models can be made into renewable resources that is of high quality for reliable use by researchers. As you know, cancer treatment, the most curative treatment is by surgery if the cancer diagnosed at early stage. But for most cancer types, depending on cancer type, there are various systemic standard cytotoxic chemotherapy and radiotherapy. And also there are cancer biomarker specific therapies. We call them targeted therapies and also immunotherapies. So how can we use the patient-derived models to study the clinical challenges in treatment of cancers? Because most cancers are detected at advanced stage where surgery treatment is not possible. The treatments stop working, and cancers are without actionable gene alterations, meaning there are no targeted therapy available. So I would describe the context of the clinical challenges and the use of these models in the study of non-small cell lung cancer. As you can see here, lung cancer is diagnosed at early and late stage of disease. After surgery, 30% of the patients do relapse, and in most cases, lung cancer is detected in later stages. Lung cancer is also classified into the histotypes. The major histotypes are the non-small cell, 80%, and the small cell. Here we will focus today on the major subtype of non-small cell, the adenocarcinoma, called LUAD for short. And they are further classified into genotype classifications for targeted therapy when possible. In reality, we know that targeted therapy, even though they are primarily identified through one driver oncogene, in which the alteration increases signaling and dependence of cancer survival, and the targeted therapy inhibits the signaling activity and thereby causing fast response to the therapy. In reality, these tumors are actually classified by a group of variants, and potentially these unique set will impact on treatment response and outcomes. We can appreciate these in such studies of population tumor profiling databases. Here we have the Cancer Genome Atlas project, where the consortium profile 11,000 tumors from patients representing 33 tumor types and 10 rare cancers. As we can see here by report by Bailey and team, in studying these 33 tumor types, do these variants impact on treatment? Here we can see treatment as categorized as approved standard of care therapies, the FDA approved therapies, in clinical trials and preclinical research. So the first cancer here is the skin cutaneous melanoma, and you can see the other cancer with the high approved standard of care therapy, targeted therapy, is the thyroid cancer, 6 columns across. Now, those two cancers are characterized by dominant mutations for most of the tumors. Whereas the other cancer types, you'll see that there are no targeted therapies approved, and the variants are still being investigated for therapeutic impact in clinical trials or preclinical cancer research. Here, I've also highlighted down there the lung adenocarcinoma, which is the focus of today. And the other major subtype, as you know, is squamous, further along there on the bar chart, where there's no approved therapy. Back to the lung adenocarcinoma, we see about 10% or just less than 10% of the tumors are characterized for use with targeted therapy. The tumors have activating mutations in the RTK-RAS-RAF pathway, with a handful of these mutations recognized: EGFR, the ALK and ROS rearrangements, and BRAF. And also, when the first-line therapy fails, the resistant tumors tend to activate alternate genes in the signaling pathway, in the RAS signaling pathway, the MET, RET, ERB2, and TRK and RAS. So how do these targeted therapy impact on patient outcome? As you can see here in the figure to the left, we have overall response rates. And on the y-axis here, we have response rate with respect to targeted therapy in the lung cancer, lung adenocarcinomas. And we can see that initial treatment response is very high. Up to 80-90% of the patients do respond very well. However, on the right panel here, we see that progression-free survival doesn't change too much, and most patients have disease progressions within 2 years. So that's one of the challenges of therapy. The other challenges, of course, is that some of the cancers lack alterations in the mitogen-related signaling genes. Most of the targeted therapies are within this class of biology, inhibiting the mitogen-related signaling genes. Here we show that in the study by Zhang and team, they show that of the adenocarcinoma, 33% in the TCGA cohort lack such alterations. Instead, these tumors have alterations in the tumor suppressor genes, as noted by the red arrows to the left. They also have variants that are complex, that changes the structures in terms of gains and losses. And at the moment, we don't know how these complex changes impact on therapy and patient outcome. So here we can see that traditionally tumors have been collected from surgery, biopsies, or fluids, pleural fluids, ascites, or blood. And as we can see, these are limited biospecimen. And studies such as the TCGA have allowed us to better understand the underlying causes of tumor aggressiveness. However, we need to understand how it impacts on function. So how do we do this? We would try to collect tumor specimens from all these clinical collections and try to make the patient-derived models. So here I'll just review quickly that traditionally we've used human cancer cell lines that are propagated for a long time on plastic dishes. Now we'll be introducing the patient-derived models, the xenografts and the organoid models, which hopefully will better represent patient tumors. I'll quickly review the advantages of the different types of tumor models. In the traditional 2D monolayer cells, we have them growing quite quickly within 2 or 3 days doubling time. They're easy to manipulate. In that way, they're very cost effective. And these cell lines have been long time study and extensive data and experience have been gathered. And we have large collections of knowledge through these resources I've named here, the CCLE, the NCI-60, GDSC2. These are data portals or collections of large-scale cell line drug screening with results, and also portals where the PharmacoDB and Synergy XDB, where we try to collect all large-scale drug screening under one portal to be searchable by variants, and also the genomic profiling. So the disadvantage of using 2D monolayer cultures is that they have high selective pressure in the plastic culture environment, and they represent poorly the genetic diversity that we see in patients. Most importantly, they are poor predictors of patient response. Compared to the PDX, patient-derived tumor xenografts, these mimic well the parent tumors. They have a tumor microenvironment representing hypoxia acidity, and they also have better explant success rates for a wide variety of tumor types compared to 2D cultures. Of course, the disadvantage of that is that there's operational experience is needed. They are very slow growing, between 2 months per cycle, serial passaging to 6 months. There's the murine stroma. They lack the immune component, and also they are difficult to genetically modify. The other models are the 3D organoid models that are appearing in the last few years. And I like to describe first the patient-derived organoid models. So we know that the organoid culture method refers to cell growing in a 3D in vitro environment that can form self-organizing structures of differentiated cells. The growth of normal small cell intestinal organoids as self-organizing crypts was first described in 2009 by Dr. Hans Cleaver's lab, from which media was then refined to establish the patient-derived tumor organoids that you see listed here. This is one of the figures in his review paper, and it lists some of the major tumor types that's been established with the highest success rates in pancreatic and colon cancer models. In the lung organoid model, this project is run by Dr. Nikki Radulovich and a former PhD, Kelly. Nikki is the Living Biobank organoid manager, and she and her team worked out the method, refining the media culture from normal lung cells to grow the patient-derived tumor organoids. So Kelly and Nikki's team received collections of tumors from resections or biopsies, and the fragments are enzymatically dissociated. And if they're from the mouse cells are depleted. The cells are resuspended in the Matrigel matrix, and media is overlaid, and the cells are grown within a couple of weeks. So Nikki lists here 1 to 3 months for 2 passages. And if they grow successfully, they will be banked, cryopreserved, and viably blanked, or else they will be expanded for drug screens using 384 Wells method. So the organoid use and descriptions and applications of them are described by Nikki previously in the last year's drug discovery virtual event series. Here I would describe some of the updates and some of the more recent uses with PDX. We can see here from the organoid inventory that the major tumor types are being represented. We have pancreatic ductal adenocarcinoma, colon, lung, small cell, esophageal, ovarian, and breast. And here, the second column, you can see the different sources of tumor cells from surgical biopsy and from the patient-derived xenografts tumors themselves. And we can see the breakdown of how many models have been successfully made. And you can see some features that Nikki's team has recorded here, that the organoid models do vary in their success rate. They have characteristics that we can take note from the passage ratio, passage duration of the passages, the doubling rate. And at the moment, the key is collecting the molecular profiling data on these models to compare back to the original tumor sources. And as you can see here on the third row, the non-small cell lung cancer, we can see that most of the models made from the PDX. We can learn from the experiences of the non-small cell lung cancer that take rate can also be dependent on the source of the tumors. In terms of how we collect these clinical sources to make the models, here we can illustrate it through the workflow for the tyrosine kinase inhibitor timeline of the patient management for lung cancer. Whether they are coming from the original surgery or if they've come onto for treatment of first line or other lines of therapies, and disease progresses, and samples need to be taken to further monitor disease and also identify what other variants are evolving to cause resistance and possibly put the patients on alternative therapies. We've worked in here that excess tissue from the tissue biopsy or liquid biopsy used for diagnostic be used for the establishment of the patient-derived models. We do try to integrate the use of these models together with clinical management strategies. For example, if the testing of the next generation sequencing panel testing comes out and the PDO drug screen has been able to be followed up in parallel, then perhaps the two information can be used to better direct patient care. But often these patient-derived models have a timeline that is much slower than the progression of the disease of the patient themselves. So they tend to be used in preclinical studies. Here I've summarized the inventory of our organoid models is near 240 models and increasing with the collaboration of our investigators at the Institute to run their research programs. For the PDX models, we have close to 600 models core banked, and also new models being established continuously from our clinical research programs that I just described previously. And for the core banking to occur, we do follow a stringent method of quality control checks for histology, cancer markers, DNA fingerprint to verify with patient tumors, pathogens panel testing, and also verify that the models could be stably passage, and also the characteristics are being monitored to verify that the models are robust for use in experiments. So here I'll move forward to describe the PDX methodology. From our own work, and some from other labs here represented, we can see that PDX is able to establish for a range of tumor types, the duodenum, the ovarian, head and neck, pancreas, lung, and colon. And here you can see a range of the established success rates. And for the most part, the large collections of the PDX have been able to establish due to the availability of the immunosuppressed mouse used as the host. And here we can see in 1995, the not-SCID variant is made available. And so a lot of the large collections has started to be established at this point onwards. At our institute, we've interchangeably used the not-SCID and the NSG, which is a triple depletion of the T cells, B cells, and the natural killer cells. For the solid tumor types, we've not seen a major difference between either of these strains, but perhaps if you're doing stem cell focus cell limiting dilution assays where it's very few cells being injected, there is some differences, whereas the NSG would be preferred strain to use. So here, what are the determinants of success for PDX? We learned this from most obviously here is from our HFR tumors, reported by Martins and team. And here we can see that for the EGFR mutants, because they are very poor at forming engraftments, larger specimens, therapy-resistant, and metastasis have a higher odds ratio of engrafting as PDX tumors. Taking a step back, looking at all the lung tumor types, we can see that engraftment on the top left panel, we can see that engraftment predicts for much shorter disease-free survival in patients compared to the non-engrafting tumors. And as a breakdown from the multivariate analysis here, we can see that squamous, advanced stage, and engraftment are all independent factors for poor disease-free survival. On a closer look, for the cohorts that we do have representation in terms of the different genotypes, we can see the EGFR cohort are very poor engrafters. And that is reflected also in the fact that patients do better in terms of outcome. Compared to the KRAS mutant tumors, you see at the lower left panel here, engraftment is fairly high for KRAS mutants compared to KRAS wild type tumors. So a lot of these determinants of PDX successes are from studies that use tumors implanted at the subcutaneous flank. Now, I'd like to quickly acknowledge that we've described that from one of our research labs here in the lower left panel for the subcutaneous flank method, but we also have PDX of other methods, for example, the orthotopic implant, where tumors are implanted at the original primary site, or we can have PDX that represents characteristics of metastasis functions, and also PDX that have an immunohumanized mouse component for drugging strategies using immunotherapy. So there are various ways to establish PDX. For our purposes, we'll focus on PDX established in the subcutaneous pocket at the flank of the mouse. Here, you can appreciate the design where you have a limited tumor fragment being implanted into the first generation of the mouse called P0. There's limited tissue, so we can only do 1 or 2 mouse implants. Once the tumors expand to a size of around 1.5 centimeter, we do harvest the tumors and serially divide them and reimplant them to generate more tumors resource, more tumor specimens for studies and for future research, where we cryobank them, in viable tissues for future research. In terms of passaging of tumors in mice, there are some quality controls that one must be aware of. There's also the spontaneous lymphoma outgrowth here shown in panel designated number 2, where you can see tumor islands with the pancytokeratin positive cells in a field of lymphoma outgrowth. So in such cases, we consider tumor model failed. Also, we review histology for potential bacteria or fungal contaminants. This can occur in oral cancers, as shown here. And those tumors tend not to be able to be propagated for a long time. In terms of study design, we can see here that once the tumor is being expanded stably, we can design a study where groups of 1 to 12 mice are used for drug testing, where we have the first group is a vehicle group, and then the conditions 1, condition 2, and so on. At our institute, we have standard operating procedures, and we use a study log mouse database to acquire all of our data and to manage and graph our data. So we use digital calipers that reads directly into our database and also digital balance. Some of the features that one can measure in terms of tumor growth here, you can see in the y-axis is tumor volume as estimated from the length and width of tumor measurements with the skin fold. And here you can see over days, typically between 30 to 60 days, growth to the maximum size of around 1.5 centimeter in largest diameter. You can appreciate that the growth rates are a little bit different between the replicates, and thus we need the replicates to monitor this variance here. And the effects of drug, for example, drug A, where we see slowdown of growth inhibition as a flat line growth here, and also the red line indicating some cytotoxic effects. Here I would direct you to a paper by Jessica Weiss from our team. She's a statistician staff at our team, and she's consolidated over 44 studies representing over 25 unique models and 15 drugs. And she addresses the questions of how do we quantify response? Do we need to increase number of replicates, increase the number of measurement, and increase duration to reliably measure response in our studies? So next I'm gonna lead you through four different studies of how we use PDX and PDO models. Here in the first study, we're using the PDX model to show ALK inhibitor resistance. Here, I'd like to start with panel number 2 here at midpoint, where we have a biopsy from a patient in July 2016. And we initiated our PDX protocol, and we were able to establish PDX and also the organoid derived from the PDX for this model. We did our first drug screen because we didn't have the 2 clinical drugs available yet, the alectinib and the lorlatinib, specific to ALK fusion rearrangement. We used a drug that is a multi-kinase drug to ALK and MET, crizotinib, down in the low panel here, directly below. You can see that there's the vehicle treated group and also very nice response, tumor growth inhibition in the crizotinib treatment group. This caused some excitement because as you can see from the clinical timeline, here the patient is resistant and has progressed from the first line of ALK targeted therapy. And so the next line of ALK targeted therapy is lorlatinib for tumors that are resistant to alectinib. So we're surprised to see some sensitivity here. So Kelly backtracked. She's our former PhD student. She backtracked and she retested the PDX using the exact 2 clinical therapies. And as you can see from the panel, the growth panel to the left, the 2 therapies are resistance in the PDX. So that tells us that the PDX is a reliable model to mimic patient resistance. And so we were curious, since the crizotinib is a multi-kinase inhibitor, the other target would be MET amplification. So we took the biopsy and we retested for MET amplification, as you can see highlighted yellow. And yes, we did verify that the biopsy had MET amplification. So as you can see here, even though clinically at that time, they were only doing single gene testing, as you know now, when tumors progress, patients are being biopsied when possible and tested with next generation sequencing for a whole panel of possible actionable genes and also known resistant mechanisms that could be targeted. So in the next study, I'd like to show you work done by Josh Rosen, our current PhD student in our lab. And he's here trying to see if potential therapies for KRAS mutant. Do they have the same effect across all KRAS mutant subtypes? As you can see here, he has a few models with the subtypes of G12D, G12C, G12V, and G13C. And here in panel figure 3B, Josh used the PDX to do the drug screen for the drug Selexinor, which inhibits a nuclear export protein, exportin, and is thought to be, KRAS mutant is thought to be sensitive to this drug. And he compared it with another drug, possibly inhibiting the same pathway, the RTK RAS-RAF pathway. And he wanted to see if the two drugs had the same effect, since potentially they're inhibiting the same pathway. Here you can see that through linear mixed modeling effect, looking at the individual mouse growth, Josh has modeled the effects of the vehicle and shown effectiveness of the two drugs of selinexor and trametinib. And here we can see that when comparing drug inhibition with the vehicle control group, that both drugs inhibit tumor growth quite well across the models. And he asked the question, does the specific subtypes of the KRAS mutation have an effect? And for models that have enough representation, we were able to do this test. And here you can see that there's no selectivity in the KRAS subtype for drug sensitivity. However, when Josh looked at the TP53 background of these tumors, it seems that P53 mutant models together with KRAS can result in less sensitivity to drug. So as we know, there are more drugs available for the KRAS mutants tumor. And because the testing using PDX is very time consuming and could take up close to a year to screen up to 10 models, Josh went on to make xenograft-derived organoids. And he showed this in one of his drug screens using specific inhibitor for the KRAS G12C. How do we show that these drugs are selective for G12C? We compare them to other types or the wild type of KRAS. And here Josh shows that with the xenograft-derived organoid models, we're able to show that the KRAS G12C, the green lines, are more sensitive to the inhibitor than the KRAS wild type models. And here they're shown in their the dose range that they were tested on. And here in the right panel by their IC50, you can see that most of the mutants are being inhibited, except there's one model that is resistant. And as for the KRAS wild type models, there's a range of sensitivity. So the interesting question is what is the underlying cause of tumor heterogeneity? And this is also observed at the clinical level. And Josh is moving forward to analyze these models, not only at the genotype level, but at the transcriptomic and also the proteomic and the phosphoproteomic level to see if there are other features that we can pick up to predict for tumor sensitivity. In the next study that I like to point to is the use of PDX to model drug-tolerant persister cells. This is a phase where cells can adapt to targeted therapy before they become resistant. Here you can see the panel to the left. We have a model with the typical activating EGFR mutation, and it's very sensitive. We have the vehicle control arm growing, and then we have the treatment on the first generation tyrosine kinase inhibitor selective for the EGFR mutant. And we can see here that the tumors actually become almost non-palpable during this phase of drug-tolerant persister. And how do we know that they are drug-tolerant? is because once we give them a drug holiday, as you can see here, with this starting the initial growth, once the drug holiday is lifted in the open circle, there are viable cells, and those cells do grow up and behave like the original tumor. So Erin, our former graduate student, did some histology and realized that in the drug-tolerant persister phase, this environment is a stromal-rich environment. And she further investigated to take sampling at different time points during this phase to do single-cell RNA sequencing. So what do we learn from that? We learned that in terms of the exomic mutations, that the drug-tolerant persister phase is very similar to baseline phase. And here from the RNA-seq analysis, the results are shown as Tisney's plots, in which each dot is a cells, and cells are placed nearby one another if they have similar transcriptomic profiles. Here you can see the dots being placed as a whole population, and we have the 137 baseline colored in red and the drug-tolerant persister phase colored in blue. And we can see that in panel A, they are clusters concentrated in one cluster. In panel B, you can see that this cluster, we termed it number 6, is a group of cells that in panel D, you can say that they are quiescent. This was not surprising, but we are in the process of looking at the expression changes and mapping the enriched pathways to determine what signaling pathways is involved in maintaining this drug-tolerant persister phase. We also realized that the murine stroma is looking different between the baseline and the drug-tolerant persister phase. Remember before I was mentioning that one of the disadvantages of PDX is the murine stroma. But in looking at the murine stroma here, we may glean some useful information that the stromal, the cancer-associated fibroblast stroma, can also be influenced or probably may influence drug-tolerant persister cells. Here we can identify the drug-tolerant persistent stroma clustering together compared to the baseline stroma. And Roya went on to elucidate what was happening in these population between the baseline and the drug-tolerant phase. Roya has elucidated some of the factors that could be secreted, the different factors to maintain proliferation at baseline phase, and also the factors that can help with cell survival during drug-tolerant phase. And this has been reported in paper by Mogal and team recently. Another result from our lung PDX studies is also what I've shown before is using mutations and transcriptomic to identify underlying causes of drug response and drug resistance. Here I will show that we have new data using proteome profiling by TMT mass spectrometry. And here we have a paper that classify our xenografts with respect to the proteotypes. As we can see here in panel A, the proteotype are identified by the groups shown in blue, the lung adenocarcinoma group 1, 2, and 3. And as you go down the rows here, we have stage being identified, various clinical features, and also the transcriptomic classical subtypes and the methylome subtypes. And we can see that the prototypes actually identify a totally different group of grouping of tumors that are different from transcriptome predictions and methylome predictions. And that the prototypes in panel C here, you can see that group 1, 2, and 3 have different overall survival for the patients. Group 3 being the most aggressive in the green line with the shortest overall survival. This is reflected in that it is representing tumor stages that are higher. In the panel E below here, you can see that in group 1, group 2, all stages of lung cancer are represented, but in group 3, that is lost. What's also interesting is that different altered genes are being enriched in the different groups. Group 1 having the best survival has the enrichment of the EGFR mutants, followed by group 2 with the various variants, and group 3. Interestingly, what do these prototypes tell in terms of drug response? We had an independent study that with having the prototypes helped us to understand better why there was drug response. We had a model called PHLC 134, and we did a pan-tyrosine kinase drug screen. And here in panel D, you can see some effect. And it responded really well to EGFR kinase inhibitor. And we were curious as to why, because it didn't have the activated mutations. Here you can see that from the genomics, we see that EGFR is amplified. And from the mass spec analysis of the proteins, total protein was much higher compared to any other lung adenocarcinomas, as you can see here plotted. And in the panel below, we can see the EGFR tyrosine phosphatidylation sites here that is being activated. Perhaps this is a feature that has resulted in drug sensitivity, but we know that in clinic, EGFR amplification is not a reliable feature for targeted therapy. As you can see here, it may have depended on protein being expressed that we see in this model. So I've hoped to describe all these 4 scenarios to you to show that PDX and PDO modeling can help us better understand some of the cancer treatment challenges. I will move on to emphasize that for these models to be renewable resources and to be available for researchers to use, they must be organized in a living biobank format. Because here you can see that there are a lot of operational efforts involved in terms of the establishment of the models and also the biobanking, which is supported by standard biobanks for the tissue itself. But for these patient-derived organoid models, they rely heavily on the accumulated data for the proteogenomics and also the clinical data that needs to be curated for proper use of these models and proper studies to be performed depending on what phase of the patient clinical management they were collected from. So I would like to emphasize that data is an important feature for living biobank and for the use of these patient-derived models. As you can see here at our institute, we are supported by the data science group to help upload all the genomics and the proteome data into our own institutional cbioportal.ca database. And here you can appreciate, here we have all the xenografts that have been profiled, including our latest edition of the organoid models. And all these studies can be integrated and analyzed together. And here I've just put a simple representation of some of the genes across our adenocarcinoma models. And you can appreciate that you can overlay the different DNA RNA and protein alterations and see perhaps what is the truly underlying cause of the phenotypic drug response. Also, I like to emphasize all the data that is collected during model establishment, which is typically not being collected by any data portals or by standard cancer banking of tissues. We have our QC data, DNA fingerprinting data that our institute has built a customized portal for us to upload all the data associated with our models to verify them with the patient tumors. And we're still working on using existing data portals or customizing our own to keep other types of data that are very important for experiments and the design of the experiments, and for QC process, such as the pathogens database that we have to monitor. In terms of making the models available to researchers and also combining our efforts, as you can see from the 4 results, once you get down from classifying the tumors as driver genes, if you go further and classify them by co-mutations, the representations of that particular genotype for study becomes very small. We're dwindling down to about 3 or 4 models of similar characteristics to test and study. So how do we combine our resources to do that? We have some of the online portals right now. We have the Cancer Models portal supported by NCI, the EuroPDX, and also some commercial vendors, where all the researchers upload the models up to this portal for visualization. And users can email directly the model contributors to access the models. And from the cancer models, of course, you can go directly to the different programs that are biobanking PDX and PDOs. For example, the NCI, which is the Patient-Derived Model Repository. We have the EuroPDX Consortium. And also, relatively new is the organoid arm from the Human Cell Atlas portal. Just to give you an idea, on the patient models portal, we have about 27 labs. They have 27 labs represented in terms of contributing data to this portal. And the models can be searched by genotype, tumor type, treatment type, and various other features. Here, I've simply listed the labs, and we, the Princess Margaret Living Biobank, is a part of this portal. Most of our published models are listed here, as you can see. And I think this is a very good way forward for having these models available to researchers, and for progressing towards studies that are of interest to precision medicine, because of each of these tumors are truly unique when we get down to all the variants that classify them. I like to re-emphasize here that there is considerable effort in the establishment of the models themselves, the technical expertise, and the tumor type specific methods. In the second box, there is also another big jump and effort, another big effort for the annotation of the model, the clinical pathological annotation, as well as the molecular and proteome annotation. And then, of course, last is the fun part. Once the models is established and annotated, we can design the experiments to address, hopefully, the clinical relevant studies to improve patient outcome. So some of the take-home messages on patient-derived models here is that we need more models. We need more models to represent the tumor diversity. We need to continue to evaluate these models for fidelity towards the original tumors and how well they continue to behave like the original tumors in terms of drug response. We also need to profile these tumors that goes beyond standard biobanking, and next generation biobanking also has to support in the data resource management. So hopefully once the models are made, we can use these models to obviously identify potential new cancer biomarkers and identify vulnerabilities in the resistance of treatment or novel drug classes to improve patient outcome. So to end my presentation, I'd like to acknowledge the Living Biobank team made up of the model contributors and the scientists that has the research program to integrate the establishment of these models. Recognize the organoid team run by Nikki Radulovic. And also we couldn't have accelerated so fast in our Living Biobank without the support of our UHN cores, our other research cores. That's helped us with the animal facility, the biobanking procedures, the histo biomarker lab, and also the data components of running such a program. And I'd like to thank our fundings, the Princess Margaret Cancer Foundation and the Ontario Research Fund. And we also like to acknowledge the cancer associations, which the PIs have received funds from, the CIHR and our Canadian Cancer Society. So with that note, I'd be happy to receive comments and questions, and I will address them in the emails. Thank you.